welcome to A Moment of Science, Please. I'm Ellis. So if this is your first time here, our podcast series introduces you to researchers and their research at U of T, its affiliated research centers, and hospitals. So today, we'd like to welcome Dr. Patricia Brubaker, Professor of Physiology at U of T. You can check out her recent paper titled Circadian Rhythms and the Gastrointestinal Tract, Relationship to Metabolism and Gut Hormones, which we have linked in our description. So let's get right to it. Dr. Brubaker, could you please start us off by introducing yourself and telling the audience a little more about you and what really interests you about the general field of physiology?
Hi, so thanks so much for inviting me to speak today, Ellis. I'm really pleased to be here. So I, as you said, am a professor in the departments of physiology and medicine at the University of Toronto. I have been at the University of Toronto for quite a few years now. I've been a professor since 1985. And yes, it feels like a lifetime sometimes. It's a wonderful lifetime. So I did my PhD studies at McGill University and really fell in love at that point in time with research as a possible career. When I went into research, I wasn't sure exactly where I was going to end up, but by the time I finished my PhD, I was in love with this notion of research and therefore did a postdoc and then continued as an academic in research. And so that's really my education. How I ended up in physiology was really an interesting sort of route. I actually started out my undergrad being interested in organic chemistry, and through that was introduced to biochemistry, which is really the organic chemistry of life, if you will. And during my PhD studies, really focused on what turned out to be a physiological system. So even though I was in the Department of Biochemistry, I was really using a physiology approach. So when I started as a faculty member, it was a logical progression for me to move into physiology, where I use a wide variety of techniques from molecular to cellular to whole animal and even occasionally human studies, all addressed at answering a question about how a system works in the whole body. So I really love the integrated nature of physiology.
Yes, your path shows how I think everything is linked together and everything has a relationship. So how did you choose your specific area of studying in endocrine and diabetes?
Equally an interesting and rather unexpected path, I guess. I initially started out in endocrinology for my PhD studies because I really loved the work that professor, my supervisor, was doing, and I was introduced to him in my undergraduate career. So when I wanted to do research, it made sense for me to approach him about doing studies in research in endocrinology. But while I was a first-year PhD student, I quite unexpectedly developed type 1 diabetes myself. And I decided, even though I was in endocrinology for research, that I wanted post-PhD to really focus on the endocrinology-specific discipline of diabetes. And so that's really how, when I came to Toronto, I started doing postdoctoral studies with Dr. Mladen Vranic, who was quite a well-known researcher in diabetes at that point in time. And then I continued those studies as an investigator with my own laboratory.
So your research is both a combination of your interest and very relevant to you. So what has led you to your specific interests in studying the factors determining tissue-specific synthesis, secretion, and bioactivities of regulatory peptides?
Right. So when I was a postdoctoral fellow, the Mladen Vranic, the man with whom I worked, was studying something that was then known as extrapancreatic glucagon. And it was a very poorly characterized substance. And in fact, while I was a postdoc, the gene was cloned. And it turned out that the gene expressed not only glucagon, but also two novel peptides, which are called glucagon-like peptide 1, GLP-1, and GLP-2. And I have, since that time, remained fascinated with the biology, if you will, of both GLP-1 and GLP-2. Because shortly after I started studying these peptides, it turned out that GLP-1 was actually your body's natural anti-diabetic peptide. And there was my link, right, from extrapancreatic glucagon now to GLP-1, which was anti-diabetic. And so I've continued to study GLP-1 throughout my entire career. And not only is it naturally anti-diabetic in your body, stimulating insulin and reducing body weight and having cardioprotective effects. So all of the things that are good, in particular for people with type 2 diabetes. But in addition, long-acting GLP-1 receptor agonists were developed in the early 1990s that meant that GLP-1 mimetics began to be used for patients with type 2 diabetes. And so there was this very exciting clinical application that happened with GLP-1. Similarly, the same story with GLP-2. I was doing some work with one of my colleagues at Mount Sinai Hospital, Dr. Daniel Drucker, and we discovered that GLP-2 stimulates intestinal growth. And again, a similar story where I was interested in the biology of GLP-2, but in fact, because of its potent growth effects in the intestine, long-acting GLP-2 receptor agonists have now been developed and implemented in the clinic for the treatment of patients who have something called short bowel syndrome, which is basically when you've had large sections of your intestine removed and you can't eat food by mouth anymore. And so what GLP-2 does in these patients is help them grow new intestines, sometimes to the point where they can stop intravenous fluids and intravenous feeding and start actually eating food and taking fluids in by mouth. So again, a very clinical translation of the very basic biology that I'm interested in for these peptides.
Yes, and I think you've also seen in person how the impact and the progression of research has proceeded over the years. So that's amazing. So I'm going to dive a little deeper into your research and into the specifics. One of your recent papers is titled The Circadian Rhythm and the Gastrointestinal Tract Relationship to Metabolism and Gut Hormones. So could you give us a brief summary as to what this paper is about?
So this paper is a review article of a field in which my lab now has a great interest, which is the circadian rhythms, specifically as related to the hormones that we're interested in, GLP-1 and GLP-2. And the review article was written by 3 very talented trainees in my laboratory, and I give them full credit for this article. So the premise behind the article, which reviews the field, and my interest in the field is that people who have circadian disruption, so for example, shift workers, who instead of being the way normal humans work, which is when you're awake during the light, you eat, and when you're asleep during the dark, you eat. And circadian shift workers are circadian disrupted because it changes that normal biology and puts them in a situation where when they should be sleeping and not eating, they are actually awake, and then that's when they're eating. And so this disrupts this normal relationship between food intake, the hormones that are secreted in response to food intake, and then if you imagine, for example, GLP-1 stimulating insulin causing deposition of those nutrients. So normally during the day when you eat a meal, you release GLP-1, it stimulates insulin, and insulin causes those nutrients to be deposited in your muscle and your fat and your liver. And all of those systems are integrated together to work in a seamless, coordinated fashion. In circadian disruption, when you're eating at night now, your body isn't optimized to take in those nutrients, release those hormones, and deposit those nutrients. And so shift workers, for example, have a much higher risk of developing obesity and type 2 diabetes because it disrupts their normal physiology for this food intake system. So we're really interested in what controls this circadian system and how food intake is linked to the release of the hormones, which are linked to the release of insulin, et cetera. And we're really in the lab studying the biology of how that system works, again, at the molecular level, the cellular level, and then at the whole animal level as well.
I think the circadian rhythm definitely interacts with a lot of different processes in our body, and endocrine is definitely really important.
Yeah, it does. And I will say that these are incredibly difficult studies to do. And I personally would never be able to stay awake all night for the studies that my really amazing trainees in the lab do. We have some studies that run for 48 straight hours, every 4 hours. These graduate students and undergrad students, lots of undergrad students and postdocs are incredibly dedicated to these studies.
Yes, I think from this kind of research, we can see the passion in people. For many of us students who haven't been in research, our idea of research is often very limited to wet labs like pipetting or roles like that. So having been in the research field for such a long time and as a senior researcher, we were hoping that you could tell us a little bit more about the different aspects of preclinical research and what you deal with on a daily basis.
Yeah, so preclinical research is what we do in the lab, right? And in large part, that what that really means is we're not testing something in humans, which would be, for example, a phase 1, 2, 3 trial of a new drug in a human, for example. So the experiments are exceedingly varied. And yes, unfortunately, a lot of it is just repeat. We do a lot of molecular analyses where you're pipetting into 384 well plates, for example. We do a lot of radioimmunoassays where you're dealing with test tubes. We do a lot of tissue morphology studies where we have students who are counting the number of proliferating cells at different times of day and night, for example, in the intestines from our animals. And again, as well, we do animal studies also at different times of day and night, looking at the normal physiology of what happens to the gut hormones when you, for example, drink a glucose solution, and then how that changes if you do it in the middle of the night, when, again, the abnormal feeding patterns, for example. And we do this in humans as well. We've done a few human studies where we've kept humans awake all night and given them food in the middle of the night as compared to food in the middle of the day. So all of these, I think, are really what we would consider to be preclinical studies. But which study requires which approach is going to vary depending on the study.
Got it. There's definitely a lot of variations between different studies. And I think I've noticed that too with research papers, reading the methodology and seeing the differences.
Yeah, I would add that I think what's really exciting about what we do in my lab is that we do something in a mouse cell model, for example, and then we try and reproduce it in human cell model. And then we go to mouse tissues and we try to reproduce our findings in human tissues. And sometimes we'll do it in rat tissues as well. And then we go to the animals and we'll do it in mice and we'll do it in rats. And then eventually we say, is it still true in humans? And so what's really nice is to being able to do this step back, big integrated picture and say that the preclinical work that we do in the lab, even the work in the cells, we can take that to an animal and we can take that up again sometimes to a human and still see the same patterns and see the same effects, which means that it's meaningful. It's not just a one-off, one cell line, but not another cell line.
I think this is really important too, to be able to take something at the molecular level, put it in animal models, and then finding this link again. And I think this is what a lot of people are fascinated by in research, to see this impact in humans. I know that you also co-authored another paper titled The Discovery of Insulin Revisited, Lessons for the Modern Era. Could you tell us a little bit more about this paper and what led you to write about this historical significance of insulin?
2021 is the 100th anniversary of the discovery of insulin by Banting and Best, McLeod, and Collop in my department, the Department of Physiology. First of all, this is a really big year for the Department of Physiology, and it led to our one and only Nobel Prize for the discovery of insulin. So it's a really exciting year in terms of being able to celebrate the discovery of insulin. A little bit of a damper on the in-house celebrations, unfortunately, because of COVID. But what has happened as a result of this centenary is that there have been lots of invitations to speak publicly about the discovery of insulin. And this paper, which was written for the Journal of Clinical Investigation, was actually an invitation to one of my colleagues, Dr. Gary Lewis at UHN, to write a review article about the history of the discovery of insulin. And so Dr. Lewis invited me to co-write this paper with him. And basically what we did in the paper is we talked a little bit about how insulin was discovered, the fact that Banting was a general surgeon who'd just come back from World War I. He was, unsuccessful in setting up his own medical practice, but he had an idea in the middle of the night. And he wrote that idea down. We still have the original notes that he made. And he thought, this is how maybe I could help cure people who have type 1 diabetes. And in 1921 and before, type 1 diabetes was a death sentence. You died if you got type 1 diabetes because you had no insulin, you had no way of depositing the nutrients that you took in your diet. And so it was horrible. It was literally a death sentence for, in particular, mostly children who, when type 1 diabetes mostly develops. And so he had this idea, and he came to the Department of Physiology because he knew Professor McLeod. Banting had done his medical degree at U of T, and he came and he said, I have an idea. And McLeod sort of said, well, you know what? It's an interesting idea. Let's see if it works. And he gave Banting a lab to work in over the course of the summer. And he hired an undergraduate student or someone who just graduated, Charles Best, to be his lab assistant. And through May and June and July, Banting and Best worked quite tirelessly trying to figure out how to collect the pancreas and make an extract that could treat high blood sugar levels in their experimental animals, which were diabetic dogs. And by July 30th, they were able to lower blood sugar levels in dogs. Now, this was a very impure preparation. Wasn't entirely reliable. It didn't always work. They tried lots of different approaches. And then McLeod in the late fall of 1921 brought in a biochemist, Collop, whose task was to improve the purification method. And so Collop started to work on that. And by January of 1922, January 23rd, specifically, they had a preparation that was sufficiently pure that not only did it lower blood sugar levels in diabetic dogs, but it actually successfully lowered blood sugar levels in the first human who was ever successfully treated with insulin, Leonard Thompson. And so going from a death sentence, Leonard Thompson lived for another 13 years. He eventually died of pneumonia. But many of the young children who were treated with insulin during that first summer of late spring and summer of 1922, many of them lived for another 50, 60, 70 years. So it really was a miracle. It was a cure from death. Now, it's not a cure for diabetes. There are other problems associated with diabetes. But at that point in time, type 1 diabetes was a death sentence and insulin prevented that death. So it's pretty exciting story.
For sure. I think sometimes these are the examples that show us how an idea can make such a huge impact by research and to create this clinical significance. And it's also special to researchers that you would celebrate an 100-year anniversary for insulin by writing a research paper. So that's definitely special too.
And of course, I have a very personal motivation, right? I would be if it wasn't for the discovery of insulin. So it's been a pretty exciting year all around on a personal level, as well as on a research level, and then on a departmental level.
Yes, that's for sure. So seeing all these different topics in literature on endocrine and metabolism, what do you think are some future topics that you might be interested in studying?
That's a really tough question because, in fact, I love what I'm doing now. And so serendipity happens. Sometimes you make an observation and your research sort of takes a turn and you go around a corner. And I would say that happened when we discovered GLP-2 and intestinal growth. I had never expected to be studying intestinal growth. So you went around that corner, and half my lab now studies intestinal growth. The other half studies diabetes. But those two topics alone, GLP-1 and GLP-2, I think still have sufficient interest to me and sufficient numbers of unresolved questions that I'm quite happy to keep asking questions about the physiology of those two hormones. They fascinate me. They are my passion. And they're enough for me, at least for the moment.
Got it. Yes. There's still definitely a lot of gaps in research that we would love to know more about and hear more about too.
Yeah, and there are always more questions. And one of the things that's really great about having a career in research is that I'm surrounded by young minds who always challenge me and say, yes, but what about, or what if, or why is that? And so we're always challenging dogma and saying, can we think about this a different way?
Mm-hmm. And I'm sure that lots of students are very interested in the same topic and would love to learn more about it. And this leads actually into my next question. Many students are interested in research positions. Do you have any tips or advice for students who are looking to gain research experience either throughout the school year or during the summer?
It's a really good question. And I give, for the Undergraduate Physiology Student Association, I actually give a seminar every year about why you might want to think about doing research and how to go about it. So I can't answer that question in a simple podcast. But so here's sort of in a nutshell, I would say, why would you think about doing research? The first is to find out if you like it, right? First and foremost, because it can be a tremendous career if it turns out to be something that you like. And it's probably better to figure out whether you like it or not earlier rather than later. Okay, so that would be the first thing. Some people have to take research as part of their course programs. So you have to do it and you find out if you like it. Research is a great way to get to know a professor really well and to get to know how a lab actually works. So you rather than just a course-based project, basically you get to explore a little bit and you get to think about the literature and how the literature informs what you're doing in the lab and vice versa. So I think it's a great experience to have. Okay, how do you do it? That's a little bit harder, especially during COVID. Okay, but the bottom line is you need to contact professors. You need to do that email professionally, courteously, grammatically, attach a copy of your resume, attach a copy of your transcript, just an unofficial screenshot of your transcript. Because if a professor is interested in you, they're going to ask for that information. So don't make the professor work, right? Give it to them right off the top. And make the letter personal and say, dear Dr. So-and-so, I'm really interested in your research because my mother has type 2 diabetes, or because you were my professor in course 30X, right? So personalize that. And then take a look at PubMed and look at a couple of the papers that they've written recently. Look at the abstracts, see what kind of research they do. Are you interested in that research? So what you want to do in a short cover letter is convince that researcher that they want to talk to you more about doing research. And that's for the summer and or for a research project in the winter. I think the approach is exactly the same.
For your lab, for example, do students usually come with an idea in mind, or is it more, I'm interested in this research, so I'm going to ask to learn more about it?
Most professors already have grant money for specific projects. So while a student can come in with ideas, it does happen. It's far more likely that you come into a lab because you're interested in the general area and then you're assigned to a specific project, usually a small part of an ongoing project. So an undergrad student might work with a graduate student or might work with a postdoctoral fellow to do a small part of their bigger project.
Okay. And would undergrad students, for example, be able to get a lot of interaction with the professor, or is it more towards the graduate students?
It really depends on the lab that you end up in. So if it's a smaller lab where the professor is in the lab all the time, then you might see a lot more of the professor. If it's a big lab or if the professor has clinical responsibilities that take them outside of the lab, then you might see a little less of the professor. So these are things to think about when you contact a professor: is what kind of experience am I going to get? Who's going to be my supervisor? Am I going to see the professor, on a daily basis or a weekly basis? Okay, so all very good questions to ask, and to think about yourself in advance. What kind of supervisor would I like to work with?
Mm-hmm. And I think that's why it's really important to be able to meet the professor before committing to a research project or a position or anything like that.
Yeah, absolutely. And even if it's just by Zoom right now, I don't know of any professor who would hire someone without having a meeting. And in fact, when I bring students into the lab, if I'm putting an undergraduate student with a graduate student, then I invite them to meet the graduate student as well, because you work together very closely. You have to like each other in order to have a successful working relationship. Personal communication is important.
Got it. So we're getting close to the end of our podcast, and we have one final question that we're asking all of our guests. So, do you have a favorite scientist, and if so, why?
I would say that I don't have a favorite scientist per se, but a few years ago, I read a book which was called Nobel Prize Women in Science. And it was about, I think at that point in time, the 9 women who had won Nobel Prizes. The number was very, very small. And out of that book, my favorite women scientists, the people for whom I had the most awe and respect, were the women who crept into classrooms and hid underneath desks because women weren't allowed to register in medical programs, for example, at university. So they had to learn by hiding underneath desks and listening to the lectures. And so I just have an enormous respect for the women who preceded me and were successful, tremendously successful in science against all odds. They were just amazing women, and they were trailbreakers, I will say, for every woman in science, and inspiration for, of course, anyone who is in science.
For sure. I think for all of us, the trails that people have broken before us has led us to where we are today. So we pay great respect to all of them to find where we are now.
Absolutely.
So with that, Dr. Brubaker, thank you so much for being here and sharing your research. So this does conclude our podcast. Thank you to everyone who has joined us. So if you found this podcast interesting or informative, please look forward to our coming episodes. Have a great day.